A very good evening, good afternoon, and good morning to all. I, Dr. Pallavi Saxena, SSC member of iLEAPS, and Dr. Samina, Project Administrator of iLEAPS, welcomes you all on the occasion of our fifth talk in iLEAPS Global Colloquium Series 2022. So iLEAPS is an integrated land and atmosphere interactions, and it is a global research project of future Earth, which provides understanding of how interacting physical physical, chemical, and biological processes transport and transform energy and matter through the land-atmosphere interface. So the prime objective of this colloquium is to introduce the scientific society with the distinguished and significant work of eminent scientists working all over the world. So it is a very bright opportunity, particularly for the early career researchers, to interact with the distinguished speakers and establish the bridge between the ideas and knowledge through this platform. So I would like to introduce our speaker for today's session is Dr. Silvana Munzi. So Dr. Silvana Munzi is a biologist working on lichens since 1997, and her work focused firstly on the use of epiphytic lichens as biomonitors of environmental quality. She participated in several biomonitoring surveys in different Italian regions. In 2006, she obtained a PhD scholarship at the University of Siena, where she developed her research on ecophysiological parameters in lichens under nitrogen stress. After that, she has been involved in several projects focused on the impact of nitrogen on lichens. In 2014 to 16, she was granted a Marie Curie Fellowship at the University of Lisbon, where she investigated the mechanism of nitrogen tolerance or sensitivity in lichens. For the following 5 years, she was assistant researcher at the same institution in the plant soil ecology group, continuing her work on lichens and on the concept of symbiosis in general, aiming at understanding the mechanism responsible for their ability to cope with environmental stresses. She is currently manager of an ERC project in the field of history of science at the University of Lisbon, Portugal. So today she will be presenting the talk on the unbearable excess of nitrogen, coping mechanism and ecophysiological responses in lichens, which will tell in brief that is about the sensitivity to the nitrogen that is currently one of the most used lichen functional trait in bioindication. But considering only that property only as a biomonitoring tool is an understatement. So different other facets of this interaction can be discussed in her talk, and she used to tell about the functioning of the lichen symbiotic association and the mechanism used to cope with environmental stress at ecological, physiological, and molecular level. So now I would be very happy to invite Dr. Silvana Munzi. Silvana Munzi, please, now the floor is yours.
Thank you very much. First of all, for the invitation. I thank the iLEAPS project, and thank you, Pallavi and Samina, for the opportunity that you give me. Can you see my screen?
Yes.
Okay, perfect. So yeah, we are going to talk about lichens and nitrogen. That is the main topic of my research. First of all, why lichens? Lichens are beautiful, and that could be enough to decide to study them. But actually, they are much more than that. They are incredible organisms. because they can tolerate very extreme condition from the desert to the pole, and even they show to be able to survive in space condition, because in an experiment in a space station, they actually were exposed to the condition of the open space in a dehydrate status, okay, with the metabolism very slow, almost to zero, but they survived because they come back on the Earth and they were perfectly fine. So they are really incredible. Lichens can live almost on every substrate. We know lichens growing on soils, on tree bark, on wood, on rocks and other lapidus material like on the roof. They can live on leaves, on mosses, but also on a series of substrates that are quite unusual for living organisms. And that's because they actually don't have a trophic relationship with the substrate. They are autotrophic organisms. The algal partner can photosynthesize. For that, they only need the substrate as a support. The traditional definition of lichens is that this is a symbiosis between a fungus and algae and or cyanobacteria. And even like that, it was complicated to study. But now we know that they are more complicated than that, because recent research found that, of course, lichens are composed by fungus and algae, but also by yeast, by bacteria, and by other organisms. So things are becoming really, really hard. This is just an example of one of the two main structures that lichen can have. It's an heteromerous lichens, where the fungal hyphas and the algal cell are divided in strata, in layers. So we have the upper cortex, composed only by the fungal partner. Then we have the algal layer that is in the optimal position to receive the light for the photosynthesis. We have the medulla that is composed by the fungus, but it's not so oppressed. The cells are not so compacted just to favor the gas exchange. Then we have a lower cortex and with some structure, rhizines, that serve to anchor the lichen to the substrate. Why nitrogen? Until some decades ago, the most phytotoxic gas, toxic for lichens, was the sulfur dioxide. But then, with the reduction of this pollutant, the main the main pollutant affecting lichens is nitrogen. Nitrogen is linked to our daily life because fertilizer for production or farming or production of energy or combustion of fossil fuel produce a reactive form of nitrogen. Because of course, when we talk about nitrogen, we are not talking about the atmospheric nitrogen, but we are talking about the reactive form of nitrogen. So oxides, nitrates, nitrates, and all the compounds. And these compounds can turn easily one into another. So that's another point that makes the study of the effect of nitrogen a bit tricky. Since nitrogen production is related to our daily life and the population is increasing, we cannot expect a reduction of nitrogen pollution in the future. On the contrary, we are expecting for an increase unless we really change our life. So, well, why are lichens and their response to nitrogen important? From the point of view of biomonitoring, because they are among the most reactive components of the ecosystem to these elements. So they can provide an early warning of nitrogen pollution and allow us to identify areas at risk. Lichens are also an important part of many ecosystems that still need to be studied and known, like the Mediterranean ecosystem. Explaining the effect of nitrogen on the ecosystem can thus provide useful information also to develop appropriate policy for environmental protection. We have an example, for example, we have an example in the critical loads and critical levels that are based also on empirical observation of lichens. Understanding the functioning, the relationship with nitrogen is also important because can provide useful information about the functioning of the lichen themselves. So that's why I want to show you today not only the biomonitoring part that is very well known, but also how studying this relationship gave me the possibility to contribute to the knowledge about lichen functioning. Lichen-nitrogen relationship. So nitrogen arrives in the atmosphere or wet deposition, dry deposition, arrive to the lichen and start a series of cellular molecular mechanisms that are translated in physiological effect. This physiological effect, of course, are translated on their turn on the ecology of the species. However, when we study traditionally, actually, the traditional study started from ecological observation because it was easy, even in the past, to make ecological observation. Then we moved to the physiological effect to study some physiological parameters in response to nitrogen, both in the microbiome and in the photobiome. And these two aspects, these two kinds of observation at the ecological and physiological level provide us with some means for biomonitoring purpose. However, lately, also the observation at cellular and molecular mechanisms are increased. And we are using this observation to understand the molecular mechanism that allow lichens to cope with or to be sensitive to environmental nitrogen pollution. About the biomonitoring method, mainly we have three different forms. At the moment, we can assess the change in the epiphytic lichen communities. This is the traditional method, actually. There are some problems because it requires a long time to happen, relatively long time. It can be a few years, but still it's a long time. And so it gives a late indication. The functional groups approach, it's a bit different. It's also requires time, but it's more specific because it takes in consideration more detailed description of the ecology of the species. So it provides more accurate information. And then we have the physiological approach that gives a more rapid response and can be used as an early warning. And we are going to see some example of it. Functional traits, maybe someone is not familiar with the concept. Functional traits are morphological, biochemical, physiological, structural, etc. Characteristic that characterize all the organism, the individual or the population, and are considered relevant for the response of the organism to the environment. Here I have some example. It's from ILEAPS, the information system of Italian lichens, and we have two different species, Xanthoria peritina and Evernia prunastri, that are the ones you will hear more mostly about in this presentation. And you can see that, for example, we have the characteristics related to the tolerance to the pH of the substrate, to solar irradiation, aridity, eutrophication, polytolerance, and also other characteristics like the altitudinal distribution. So when we know enough about the species to make some index, we can use the functional traits approach. For example, Xanthoria peritina is a cosmopolitan and neutrophytic species, very well adapted also in urban environment and apparently favored by eutrophication. Evernia prunastri, on the contrary, is an acidophytic lichen and tends to disappear when the pollution, the nitrogen pollution increases. And here you can see it's a study where we studied along an ammonia gradient. It was the distance from a cattle barn, so it was ammonia related to farming. And you can see that when the atmospheric ammonia increases, you have a decrease of the Evernia prunastri and an increase of the frequency of Xanthoria peretina. So it's a very clear example of the functional approach. We have a parameters, we have a factor, in this case the availability of atmospheric ammonia, and we have two species belonging to different functional groups that react in a very different way. How can we use this information for biomonitoring? It's very simple, because traditionally the idea was that if you have many lichens, the air quality is good. Actually, it's not like that, because if we have all species that are very tolerant to pollution, we cannot say that the air quality is good. This is an example. It's a study around a municipal solid waste incinerator in the south of Italy, and we recorded the presence of the non-nitrophilous species and the nitrophilous species. So you can see that when we consider the index of lichen diversity of non-nitrophilous species, we have the highest value. Sorry, I don't know if you can see my, okay, the arrow. So we have the industrial area with the solid waste incinerator, and we have the highest concentration of non-nitrophilous species in this area that is distant from the industrial area. But when we look at the nitrophytic lichens, so the species that are more tolerant to pollution and to nitrogen pollution, but to pollution in general, we have the highest concentration, the highest frequency of these species near the industrial area. So that explains quite well why we need to consider the functional group of the lichens and not the total diversity. Yeah, and that the conclusion is that we need to consider the functional traits. Now, if we look at the physiological parameters, there is one parameter that is commonly used as an overall indication of health and health status of the photosynthetic organism. And this is the fluorescence of the chlorophyll A. When we have a photosynthetic organism, this organism receives the energy, and this energy used, of course, for photosynthesis is reflected partially as fluorescence and is dissipated as heat. When we consider a lichen, we consider that the heat is not relevant because the temperature of the lichens doesn't change a lot. If we put the lichens in the dark, we have that all the center, the photosynthetic center, go in a status that is not active. So we basically eliminate the part related to the photosynthesis. So we have that all the energy received by the organism is transformed in fluorescence. And we can measure this fluorescence. When we measure this fluorescence, we have a parameter that is the FV to FM value, where FV is the variable fluorescence that is given by difference from the maximum fluorescence and the basal fluorescence. It means that when we have an organism dark adapted, the fluorescence go to a minimum level, that is F0. When the organism receive a light pulse, this fluorescence goes to the maximum level, okay? So this is the fluorescence that we are going to consider. And the parameter is the fluorescence, the variable fluorescence on the maximum fluorescence. So this is the parameter that we are going to use, and it's an indication of the health status of the organism. We use often this to measure the response to a particular stress. In this experiment, it was a medium-term exposure to nitrogen, always considering the two species that I talked about, Vernia prunastri, the sensitive one, and Xanthoria peritina, the tolerant one. The lichens were sprayed for several weeks with a nitrogen solution, ammonium nitrate and sorry, I can't see. Ammonium nitrate. Oops. Okay, several compounds with different concentration, and basically what you can see is that the green and the blue one were the lowest concentration, while the yellow and the pink were the highest. And we have again the difference in the response between the two functional groups, because we have Evernia prunastri that is sensitive also to the lowest concentration, while Xanthoria peritina basically doesn't change the FV to FM parameters with the lowest concentration, but only with the highest concentration. So once again, we see at physiological level the difference that we know between the two functional groups. We have other physiological parameters that can be considered for the biomonitoring purpose, and one is the membrane integrity. When we put a lichen in water, if the membrane of the cells are damaged, we have a leakage of electrolytes. It means that the water in which the lichens are became able to conduct electricity. So this is a simple measure that we made after a nitrogen stress to the lichens, and we saw that increasing the concentration, here you have, sorry, an increasing concentration, 1, 2, 3, 4 different concentration. This is the lowest and this is the highest, with three different with different nitrogen compounds, so potassium nitrate, ammonium nitrate, and ammonium sulfate. So we have that increasing the concentration and depending on the compounds, we have an increase in the membrane integrity. So until now, we have two parameters that we can use for biomonitoring purpose, the membrane integrity and the chlorophyll fluorescence. And here is how we use that parameters in the field, because it was around the incinerator. You have here, okay, the control and the measuring lichens that were exposed around the incinerator for several months. And we have the chlorophyll fluorescence that in this case you see didn't change, and the electrical conductivity that actually changed, increased. So that showed, this result showed that we had not an impairment in the photobiome, because of course when we are measuring the chlorophyll, the chlorophyll fluorescence, we are talking about the algal partner, but we had an impairment in the cell membrane, and that's likely linked to the fungal partner, because around 90% of the biomass of a lichens is composed by the fungal partner. Okay, so we have two different parameters that we can use, and not always we have the same response from them. So that's interesting. Then we decide, since the previous experiment were used just to identify suitable parameters, we use a very high concentration of nitrogen. In this experiment, we decide to use ecologically relevant concentration of nitrogen, meaning concentration that more or less you can find in a real situation. The experiment was the same. They were sprayed with nitrogen compounds for several weeks, different concentration, and the same parameter that I showed you, the FV to FM parameters and the membrane damage were measured, and also the performance index. The performance index is another parameter linked to the fluorescence of the chlorophyll A, and once again is an overall index of the healthy status of the lichens. And here you can see that after several weeks, we actually had an answer from the lichen also with ecological, ecologically relevant concentration, because after 2 weeks with the highest concentration, but also after 3 weeks with the other concentration, we have a decrease in the fluorescence parameters respect to the control. In this case, the performance index was even more sensitive than the FV to FM, because already after 1 week, we had an answer. The membrane damage actually didn't change, so that's again the indication that it really depends on the situation, and we really need to choose the parameters that we are considering very wisely, because not always all the parameters respond to the same stress. That was mainly linked to the biomonitoring purpose, but we also decided that we wanted to investigate how lichens works. So actually, what's defining the tolerance or the sensitive of lichens to nitrogen? We decided to test the polyamines. Polyamines are low molecular aliphatic amines. They are ubiquitous, meaning that they can be found in any organism. They can be found in free form or conjugated to other small molecules, and it's thought that they affect at physiological pH is like the stabilization of some molecules in the cell, because they are positively charged at physiological level, physiological pH, sorry. And so they can link, for example, to the DNA and stabilize it. In any case, polyamines are involved in many different processes in higher plants, the development, growth, but also interaction between the plants and the fungi, for example, or the plant and the viruses. And they are also known to be responsible for response to abiotic stressor. Ozone, osmotic stress, salt deficient, hypoxia, UV radiation. So what was our idea? Our idea was that polyamines can be also responsible for tolerance to nitrogen in lichens. We selected then 3 different polyamines and 3 different polyamines inhibitors. The working hypothesis was that if polyamines are responsible for the lichen tolerance, it means that if I give inhibitors to a tolerant lichens, it should behave like a sensitive one. And if I give polyamines to a sensitive lichens, it should behave like a tolerant one. And actually, that's really what happens, because Xanthoria peretina, that is a tolerant species, treated with polyamines inhibitors and then exposed to nitrogen, show a decrease in the fluorescence parameters, meaning that it was not more tolerant, but it gained a sensitive to pollution, nitrogen pollution. On the other side, Evernia prunastri, that is a sensitive species, treated with polyamines and then exposed to nitrogen, became tolerant. And we can see the difference, because this is the control, now treated with polyamines, and then with the polyamines, although a different time from the treatment, it became a tolerant species, let's say like that. So polyamines can be involved in the tolerance of lichen to nitrogen. Another experiment that we did was to investigate the role of chitin and ergosterol in the nitrogen tolerance. Why? Because chitin is a constituent of the fungal cell and actually contains nitrogen. Ergosterol is the constituent of the fungal plasma membrane, and it doesn't contain nitrogen, but the two are usually used as markers of the fungal biomass and activity. In an agricultural site, it's a semi-natural environment where we have two different areas, one with grazing and with a cattle farm, and one without grazing, where the cows, where the animals were excluded since 1998, we collected 4 different species. 2 species, Xanthoria peretina and Palmotrema evolucinum, we were able to collect in both the sites, so with the low nitrogen and with the high nitrogen availability. And 2 species, Severnia pronastrid and Usnea, we were able to collect only in the clean site because they are sensitive species. And we measure in all the species the amount of chitin and ergosterol. Well, ergosterol, okay, give a significant pattern, but actually the difference was not so relevant between the different species and between the same species in the 2 sites with different nitrogen availability. Chitin was more interesting because we saw that in the 2 sensitive species, the amount of chitin was the lowest. In the intermediate species, talking about the tolerance, that is Palmotrema evolucinum, there was a medium amount of chitin, while with the most tolerant, the Xanthoria peretina, the chitin was the highest. The concentration of chitin in the tissue was the highest. And not only in the same species, the chitin was highest in the site with the highest availability of nitrogen. So our idea is that chitin actually can work as a storage of non-toxic form of nitrogen. And this is one further mechanism that could explain the nitrogen tolerance in this species. That is a series of experiments carried out at the CEH in Edinburgh, where there is the Wimbox site. It is an experimental site where you have nitrogen manipulation, both dry treatment and wet treatment. And it's really interesting because actually the treatment are given in very specific condition using the rainwater, and only when there is a certain kind of climatic condition. So when you carry on an experiment there, you are sure that all the conditions are very similar to the natural one, and that the only different factor is the nitrogen. So it's a very, very nice experimental site. Okay. This is the distribution of the different plot, but what I did was to collect Cladonia portentosa. Cladonia portentosa is a soil lichen typical in the bog, so it was there in the plot, only in the wet treatment plot, because in the dry treatment plot it was completely disappeared. But in any case, I collect the lichens, and you can see also a morphological level that there are some difference between the untreated lichens and the treated lichens, because the untreated, you can see they are very long, they are a light color, but if you look at the treated one, you can see that they're shorter, and they show an outgrowth of algal cell on the surface. Okay. So there are some consequence also at morphological level. But we didn't want to only see at morphological level, so we went to investigate the algal partner, in this case, the ultrastructure of the algal cell. In the center, you can see the control. It's a very nice representation of the algal cell with the pyrenoid, the thylakoid, and everything like in a book text, really. But if you look at the other picture that represent the same species in the treatment, you can see that there are a series of change. We have the pyrenoglobules low electron dense. We have the thylakoid clustered, and they are randomly distributed. We have the accumulation of electron dense material, the wall that is really thick. So there are some change at the ultrastructural level. Are these changes related to nitrogen? Yes, of course they are, but in any case, they are not a specific answer to nitrogen, because the same kind of changes were observed also, for example, in case of pollution with heavy metals. So this is the response of the algal partner, but is not specifically linked to nitrogen. Another experiment, always in the WIMBOG, was to the analysis of the proteome, because we wanted to understand a bit also at molecular level what was the response to long-term treatment. So we analyzed the lichens exposed to different forms, ammonium and nitrate, to 3 different doses, 16, 32, and 64 kilograms of nitrogen per hectare per year. And we compared the cumulative exposure of 6 months, the short term, and 11 years, the long terms. Very briefly, the most relevant changes were observed in the fungal partner, and specifically with the nitrate, the mostly affected was the energy metabolism, while in case of the ammonium, the most affected was, beside the energetic metabolism, also the transport and the translation and post-production of the protein. That's really interesting, because we saw that the molecular response could explain the ecophysiological observation made on the species. For example, we saw at molecular response that the photobiont showed the upregulation of the energetic metabolism and the photosynthesis. And that was in agreement with the fact, with the ultrastructure that you saw, that also, in case of change, the chlorophyll phosphorescence parameter didn't show any impairment of the algal functionality. So, although there were some changes, they were not so drastic, they were not so serious to avoid the functioning of the algal partner. Then we observed that majority of the under or overexpressed proteins belong to the fungal partner and are involved in the protein transportation and regulation. That makes perfect sense when we think that the lichen species that share the same photobiont can have different response to nitrogen, different tolerance degree. And the molecular response showed that actually is the fungal partner that it is responding to the nitrogen pollution and not the algal partner. Then we saw, as I told you, we analyzed 3 different doses of nitrogen, and we observed that several proteins showed a bell-shaped curve with high expression at the intermediate concentration. And this is in agreement with the fertilizing effect that can have the lower concentration of nitrogen, and that was observed in several past experiments, and also that the repeated exposure can exacerbate the effect of nitrogen. So there should be a threshold behind which there are some change also in the molecular response. Finally, the upregulation of the enzymes involved in the protein degradation were observed in the case of the ammonium exposure to ammonium, but not to nitrate. And this is observed also the ecophysiological level because the ammonium is known to be more harmful than the nitrate in the case of lichens. In case of the dry treatment, we tested different lichens because until now I talk about Cladonia portentosa, but we also checked the Evernia prunassia and Xatoria peritina, the same that I showed before, and they were exposed in the dry treatment, so exposed to ammonia. The response was actually an early decrease in the fluorescence parameters for Evernia prunastri and a late decrease of the fluorescence parameter in Xanthoria peretina. But in any case, they both decrease. So the idea that Xanthoria peretina is a lichen that loves nitrogen is not completely correct, because we have the same response, although at different nitrogen levels. So what really changed is not the behavior, it's only the threshold of the lichen. And just the conclusion for that is that there is no linear relationship between the nitrogen availability and the response of the species, and that we need to consider the threshold, because there's a limit also for the tolerant species. Okay, I have some other things, but I think my time is finishing, so I want just to show you possible use of lichens for the response of lichens for wildlife conservation. I was invited to participate in a project in China with in a Chinese project. It was in Shennongjia in the Hubei province because it's an important natural site where the Rhinopithecus uroxellana, that is an endemic monkey species, lives. During the winter, these monkeys basically survive thanks to lichens, a bit like the reindeer here in Europe. So they used to eat lichens mostly. And the study was just to understand how the increasing nitrogen pollution linked to the development of that area could affect the lichens that would affect the survival of this monkey. And we just repeated the experiment, exposing the lichens to the nitrogen, and we actually saw that also the species that are there, that was Usnea longissima, Usnea betulina, Ramalina calicaris, and Parmodrama evolucinum, responded in the same way, decreasing their vitality in the presence of nitrogen. And not only, also the thallus growth and survival were reduced, increasing the concentration of the nitrogen doses, and also the survival of the propagules decreased. So this is just an example of how this lichen nitrogen relationship can be used in other aspects linked to conservation. And thank you.
Thank you, Silvana. The participants, you can please post your questions in the chat box. Unfortunately, in this GoToWebinar platform, you are all muted. All the participants are muted, and I'll read it out for Silvana. I don't see any questions there. Looks like your talk was very clear.
Yeah.
It
Okay.
was really, yeah, it was really exciting because I don't come from that that biology field, but it was very clear. Still, it was very clear and just self-explanatory. You did it really
I
well.
hope so. The idea was to give just a clue about the fact that it's not only important for biomonitoring purpose. Of course, it's crucial because they are lichens are among the best bioindicators, but it is not only in this sense that I used lichens. The idea was to contribute also to the knowledge on this organism. And basically all that I show is published, so is someone is interested, it can be easily found on or you can email me and I will give you. I have other work also, and I have not time. I prefer to respect the time and to have questions, but there are no questions, so perfect.
Yeah, no, there are a few questions showed up now.
Okay.
Yeah, so the first question is from Saurabh Sonwani, who is an iLEAPS ECSN representative from the South Asian region. His question is, I'm a bit curious to know that some studies mention that the elevated CO2 level can affect lichen frequencies and diversity distributions. So can we consider lichen as indicators of changing climate? Oh, interesting.
You can consider definitely lichens as indicators of changing climate because, as I show you, there are different functional aspects that we can consider. And for example, there is a study by Matos and Etalias that consider, for example, the presence of lichens with trentipolioid algal partner, because the trentipolia is an algal that enter in the symbiosis, but especially in tropical lichens, the majority is in tropical lichens. So the increase in Portugal that they observed was put in correlation with the increase of the hours of with increasing of the humidity in some condition. Okay, so you can really use them because if you have a difference in the temperature or in the rain pattern, you will observe that in the lichens. So definitely they can be used for climate change.
Thank you. Next question is from, excuse me if I'm pronouncing it wrong, the name, because it's from David Romain Arboleda. I have read once that nitrophilic lichens and non-nitrophilic lichens are affected by the pH of the tree cortex. Does this affect its biomonitoring?
Yes, it can, because, for example, when we use the defined protocol for biomonitoring, we are considering trees, because we talk about epiphytic lichens. We are, for example, considering trees of a certain age, because the pH can change along the life of a tree depending on their age. And of course, pH is relevant for the lichen colonization. But when you are analyzing a certain area, you have that the contribution of the meaning an area with an impact from pollution, the contribution it's of the pollution, it's so higher than the pH of the cortex that you actually are looking at the effect of the pollution.
He has one more question associated. When the tree cortex pH can vary because of the nitrogen contamination.
Sorry, can you repeat? When the tree cortex pH
When
Yeah.
can variate because of the nitrogen contamination? I think you already answered it.
Yeah, I think.
Yeah.
I have some observation with the pH, for example, and actually it was a high polluted area, but the pH wasn't changed so much, and the lichen colonization was changing. So it was not related to the pH, but it was more related to the pollution. I'm not saying that you cannot find situation where the pH is the dominant. For example, there are some studies that show a difference between the trunk and the branches of the trees. The colonization is different and the pH is different. But in my experience, at least where I made the surveys, the pH was not a so relevant parameter.
Okay, yeah, thank you. So I don't see any other questions from the participants. In that case, maybe Pallavi has something.
Yeah. Silvana, I want to ask that my first question, what is the mechanism of accumulation of nitrogen and particulate trapping in lichens?
Consider that lichens don't have a cuticle like plants, so they just absorb anything from the atmosphere, and they also don't have a way to eliminate this accumulation, so they just absorb and take everything. In the case of the nitrogen, there was a correlation with the cation exchange capacity. So some researchers observed that the higher the cation exchange capacity, the higher they could absorb the nitrogen compounds, mostly because they were the ions that could link to this site, and they can yeah, basically they absorb anything and they just keep until it reaches a level that is toxic for them.
Okay, thank you. And I have actually came across some of the articles on lichens act as bioindicators. So what I found that epiphytic lichens are more preferable for the bioindicator study. So what is special about that epiphytic lichens?
Because, for example, when you talk about lichens growing on rocks, the composition of the rock can be very different. They can have a chemical interaction with the pollutants that are in the atmosphere, and there can be also a buffer effect in relation to the pollutant effect. So that's why mainly we don't use it for biomonitoring. I mean, although there are differences between different barks in different trees, it's still very limited respect to what can change and what the substrate, a rock substrate can do on lichens.
Thank you. Thank you for the nice explanation.
Yeah, maybe you can conclude the session, Pallavi, if there are no other questions.
Okay. So now we come to the end of the session, and before I officially declare the session to be closed, I wish to summarize some of the important points of the talk of Dr. Silvana Munzi. In her talk, she have mentioned several aspects of lichens, particularly that is lichen occurrence in normal and extreme environment, and explain the symbiosis between algae and fungi with their morphological feature. She has also mentions about the relation between the nitrogen and lichen and explained the responses of lichens towards atmospheric nitrogen. She also explained that while lichens are efficient as ecological indicators of pollution, and helps in the biomonitoring method, and also explained clearly about the lichen's functional traits. She also talked about the photosynthetic efficiency of lichens with reference to different fluorescence, and explained the physiological parameters of different lichen species. She also explains the functionality of lichens and explained the role of polyamine in nitrogen tolerance in lichens. She also explains about the inter and intraspecific variation of chitin and ergosterol along a nitrogen gradient. And then she explains about her wind experiment site and explain about the dry and wet treatment of lichens. So I and Samina are very much thankful for a very enthralling talk on the lichens, because that's a very new topic, and very few people have worked across only on this topic, because lichens act as bioindicators, and I think they have a very important role if we keep them in the sustainable development goal actions. So in the last, I would like to thank to all the attendees for their active participation, and last but not the least, the organizing committee, the iLEAPS, who have given me the opportunity to organize this colloquium along with seminar. Thank you very much.
Silvana, there is one last question showed up now, if you don't mind to answer. That's from Dr. Dushyant Rathore. His question is, how can we estimate the pollution with the help of lichens in city?
There are many papers about it. Actually, in city you have you need to be careful, because in city you have two different things acting on lichen colonization. One is the pollution, one is the heat island effect, because very reduced humidity also affect the lichens. But there are standard protocols that allow you to measure the lichen diversity, and this functional approach to put in relation with the pollution that you have in the city. But there are other methods. For example, you can use bioaccumulation, transplanting, for example, lichens for 3-6 months in some areas. You can then collect and analyze the elements that accumulated during this time in lichens. So this is another aspect that you can consider and that can be useful in urban environment. And if you're lucky, and if the city has already a monitoring system based on the chemical or in the automatic advice, you can you can also put in the relation the exact content of pollutant in the atmosphere with the lichens. So there are really several ways of using lichens.
Thank you. No other questions, and yeah, then we can wind up the session. Thank you very much, Silvana.
Thank you very much. Thank you. It was a pleasure. Thank you. Bye.
Yeah.
Bye.
Thank you. Bye.